now. So
okay, great.
10:30 you can start.
I think it is 10:30, so I'll go ahead.
Sure.
All right. Hello, everyone. Let's get started. I'm Shikha Laloria from the Department of Biochemistry, IISc. I am your host for this webinar today. On behalf of the faculty members and students of the Department of Biochemistry, I welcome you all to today's lecture by Professor Douglas Koshland. I welcome Professor Doug Koshland from the University of California, Berkeley, to this gathering of alumni, members, friends, and colleagues of the Department of Biochemistry, and also members of our institute. Thank you so much, Doug, for accepting our invitation to speak at this event, celebrating 100 years of our department. This department was established in February 1921 and has been carrying out research in biochemistry and molecular biology over the years, and has also served as a focal point for training and for establishment of other departments and institutions elsewhere over the years. Although the speaker needs no introduction, it's my privilege today to introduce the speaker. Professor Douglas Koshland is the Richard and Rhoda Goldman Distinguished Chair in Biological Sciences and the Professor of Genetics and Genomics and Development at UC Berkeley. He's also a Professor of Molecular and Cell Biology at UC Berkeley. He's also the President of iBiology currently. Doug did his BA in Chemistry at Haverford College and his PhD from MIT in Biology with David Botstein. He did his postdoc from the University of Washington, Seattle with Lee Hartwell, and another postdoc from UCSF with Mark Kirshner. He then started his career as a staff member at the Department of Embryology, Carnegie Institution of Washington in Baltimore. And he was there for several years, and we overlapped there since I was a postdoc in his lab for some time. His lab has made several pioneering contributions towards identifying determinants of higher order chromosome organization, the famous SMC complexes, such as cohesin, and its impact on chromosome transactions. And he has been using the budding yeast as a model system for all of these studies. Those were indeed exciting times in the field, and his lab continues to make significant contributions even today, and he'll be talking about exciting work from his lab to us today. Doug has also received several honors. I'll just name a few of them because the list is very long. For example, the Helen Hay Whitney Fellowship. He's also been an investigator of the Howard Hughes Medical Institute. He's a member of the National Academy of Sciences, USA, and he has received the Lifetime Fellow recognition of the American Society for Cell Biology. He has received the Lee Hartwell Award of the Genetic Society of America. And most importantly, he has advised many PhD students and postdocs, many of whom are established investigators or research scientists all over the world. Let me also welcome all of our alumni who have joined from different parts of India and also from other parts of the world. I welcome all our guests, distinguished scientists from all over India and abroad, and these include yeast and chromosome biologists and students from other institutions. I welcome our faculty members and students from IISc who have joined to listen to this lecture this morning. Now, we have two channels, so a lot of people are connected via Zoom as well as YouTube. And before we proceed, I wish to mention a few points for smooth operation of today's lecture. So to conserve bandwidth, the audio is muted. So if in case it's not, please everybody mute your audio. And I also request all the participants to turn off the video while the lecture is going on. And if you have any questions during the course of the lecture, please type these questions during the talk in the chat box, I think addressed to volunteers 1, 2, and 3. A team of faculty and students have helped me in organizing this event, and I thank them for their efforts. A committee of student volunteers will be selecting the questions from both the channels and relaying it to me, and all these questions will be addressed at the end of the lecture. So without further delay, I invite Doug to deliver his lecture entitled Chromosome Structure is Like Mist on Eyeglasses, Obscures Vision. Over to you, Doug.
Okay, thank you. So it's a great honor to be able to give this lecture. It's I guess a good feature of Zoom that we can do it at a long distance. It lacks a little bit of a personal touch. I wish I was there. As one little way to add a personal touch, you may in the background hear my 16-year-old dog bark occasionally, but hopefully the dog will be under control. So this is the title, which is a bit heretical, because what we think is, we've all been told, a fundamental premise is structure drives function in biology. And we see that by, we see a bird with wings and that allows it to fly. The unusual structure of the eye allows us to see. The active site of enzymes, this explains their biochemical functions. And probably the most profound example is the structure of the double helix, which tells us how DNA replicates and encodes information. And so what I want to do today in my outline of my talk is really going to be in 3 parts. I'm going to try and pose that structure is a double-edged sword. Structure can be a distraction, structure can be incomplete, and structure can actually be irrelevant. So the goals of the talk really is to hopefully you'll learn something about chromosome structure by the end. And also you'll have maybe a nuanced, provocative view about the relationship between structure and function in biology. And finally, I think it's just, if you want to do creative genetic experiments, you should look to the great classics that will allow you to do something with CRISPR. That really the foundation for doing creative genetics is there. The technology just makes it more available today. So structure as a distraction, I point you to a Rubik's Cube, where you look at the outside pattern of the Rubik's Cube, and you would argue that by the outside appearance, these are very different. But we know, in fact, that we can go from one structure, this one on the left to the right, by a few simple twists of the Rubik's Cube. So here's an historical perspective on structure as a distraction. Oops. So in the pre-1980s, what we knew was the structure of cells, organisms, genomes were so different that the way you could generate that diversity, only way you could do that was obviously by inventing many new parts, which would explain why the human genome, which is much more complex than the yeast genome, is because you had to encode all these extra parts. So yeast secretion couldn't be anything like what happens in the neurons, or the embryogenesis in flies, which obviously a fly looks so different, had to be totally different from the embryogenesis in humans, thereby invoking the concept that there had to be lots of parts. And what happened in the 1980s, when I began as a graduate student postdoc, was in fact about a decade, at least 15 years of publications over and over again, which you could get in any of the vanity journals we like to call them these days, Cell, Nature, or Science, simply by discovering not a gene, the first person to discover a gene, but the second person to discover that gene, because you show that that gene wasn't present in the organism where it was originally found, but was present in another organ. So for example, the Sec genes were found in yeast for secretion were actually the same genes that were used in neurogenesis. Similarly, the genes in Drosophila fly embryogenesis were the same genes that were used in human embryogenesis. And that just people couldn't quite get the message that the way that we generate different structures in biology is not by inventing new parts in general, but actually by using a different combination of parts. So in this case, we were sort of fooled by the difference in structures, when in fact they were intimately related, just sort of sometimes rather subtle differences in how you use the parts. So in 1990, the question was, what are the factors that promote higher order chromosome structure? And what we knew in interphase that there was chromatin, which was dispersed, and that in mitosis, the chromosomes developed a very specific structure in which the two replicated DNA molecules, which happened during S phase, were actually tethered together to generate what we call sister chromatid cohesion. The second feature, of course, was that this dispersed chromatin became compacted or condensed. And so what we want to know was what were the factors that were involved there? And that was a big mystery in any organism. So if you're trying to figure out what the factors are involved in a process, you have to pick an organism. And so what's a good model to study higher order chromosome structure? Well, you can look at the 10 vertebrate chromosomes, mitotic chromosomes of a vertebrate, shown on the left, stained with a fluorescent dye like DAPI. And then you can look at the same fluorescent chromosomes of a budding yeast, all 16 chromosomes. And right here, you can see the sister chromatids, and you can see the compacted DNA of each chromosome. When you look at the 16 chromosomes of budding yeast, they didn't look anything like what you see in mammalian cells. And you think, well, maybe this is a problem with amplification. So you can amplify these things. So I like to say this looks like a schmutz. And if you amplify it a lot more, it just looks like a bigger schmutz. So a lot of people thought that yeast couldn't possibly be a good model for studying chromosome structure in mammalian cells. But what we knew was that, at least by genetically, yeast chromosomes behave just like chromosomes in plants or humans. So maybe there was something misleading about this. And so what happened was Vincent Guacci, a postdoc in my laboratory, what he did was he came and we used the technique of fluorescent in situ hybridization to look at individual chromosomal sequences in yeast. And what you see on the left here are the mitotic chromosomes of budding yeast, stained in red, the sort of schmutz I was talking about before. But now what we've done is use a probe to look at a specific sequence on a particular chromosome. And of course, there are 2 copies of that chromosome inside the cell because they've undergone DNA replication. But what you see is a single fluorescent spot, which indicates that the 2 replicated molecules have to be very close together, or in fact, there is sister chromatin cohesion in budding yeast. Similarly, if you look at a particular locus, the repetitive locus or the rDNA locus in budding yeast, in G1, what you see is the dispersed mass of the stuff. So it's spread out like you would expect in a mammalian cell. And that's been stained in green here by the dye. And now what happens, what you can see is when you go into mitosis, this dispersed mass forms a very formal structure, a linear structure, much like mitotic chromosomes in vertebrates. So now we have the same kind of structures in yeast that you have in mammalian cells when we use the right kind of way to view them. And so with that assay, we can now begin to look for gene products that were involved with that. And Alex Strunikov came to my laboratory and discovered the so-called SMC family of proteins. And these, we found out quickly, were conserved in all eukaryotes. And that was very exciting. What actually made it, I think, even more exciting was these SMC family proteins were actually found in prokaryotes as well. So what we now found out was these family of proteins were, in fact, a fundamental unit of chromosome structure in what you think in all living organisms. So once these proteins were identified, it turned out that they didn't act alone and that they acted with auxiliary factors, which are also highly conserved. And there turns out to be only about 3 or 4 of these types of complexes in all cells. So one of those types of complexes was called cohesin, shown here on the left. You can see there's a dimer of the 2 SMC subunits, and then there's these auxiliary subunits. And what that complex was shown to do in my laboratory and others was to, in fact, hold the sister chromatids together. Work really pioneered by Datsu Hirano, and then in my laboratory, we showed there's a second complex called condensin, which helps to condense the chromosomes. Okay, so now I want to tell you a recent story of structure as a distraction. So we return to here, where we're looking at differences in structure suggests that chromatin compaction in yeast and vertebrates are very different. So again, well, yeah, maybe they have these components, but if you look at the compacted vertebrate chromosome, the compacted yeast chromosome, this doesn't look very compact at all. And I told you before, you got to get this line-like structure, which one could say is pushing it to see that they're relevant to each other. Now, one of the things we knew was that the formation of this condensed structure depended on condensin, as did the condensation of, for example, the rDNA yeast. However, it didn't look that similar. And one of the things that sort of pointed out that maybe they really weren't going by the same kind of mechanism was that in the mammalian cell, this formation of the condensed structure was cohesin independent, which made sense because you use cohesin to make cohesion. and you use condensin to make condensation. But in budding yeast, in fact, condensation required both cohesin and condensin. So that seemed a bit odd. However, and what I'm going to convince you in the next part of this little vignette is, in fact, the structural similarity between what you're seeing here and here is right there in front of you once you know how to look at it. And so this work was done, and what it's going to tell us, I think, in the end, is a fundamental feature about how condensation is spatially organized on a chromosome. And it was really the primary work of a fantastic graduate student, Rebecca Lamoth, building on a work done by a previous postdoc, Brie Lavoie, and she was helped by a current postdoc, Lorenzo Costatina, and this paper has just recently come out in Genes in Development. Okay, so what we knew was that here we have the decondensed ribosomal locus in G1. In wild type cells, when you go to M phase, the chromosomes get condensed. And what I just told you, if you deplete these cells of condensin and allow them to go to this point of cell cycle so that they've never had condensin, the chromosome RDNA, the ribosomal DNA is decondensed. And similarly, you can see the same thing for cohesin. So this was the surprising result. So the question is, well, why is cohesin required for this reaction? And the inspiration to get some insight into how to think about this problem came from thinking about a very old experiment done by Lee Harwell in one of the papers that led to his Nobel Prize. So what he did in these experiments is what he called reciprocal shifts. He removed a gene product and then added it back under different conditions. So I think this is a very old experiment that is not very used very often. So in most studies you look, whether it be mammalian cells, yeast, or flies, you use conditional alleles to remove a function at a time or place during a certain time in embryogenesis, et cetera. But very few people actually take the effort to add the function back again and see what happens. So let's see what happens when you use this simple trick, what you can learn about the function of a protein. So in this experiment on the top, what we've done is we've allowed cells to go to G1 to M phase without condensin, and we get the decondensed RDNA as you see. If we then add the function back while they're in mitosis, what we can see is the chromosome condense into this line-like structure. So it's a reversibly unfolded state here that when you add the front protein back can fold. What you see when you have cohesin depleted is that when you add the cohesin back, in fact, the RDNA never folds up. So this unfolded state is different from this unfolded state because it's in an irreversibly misfolded state. All right, so now we've already learned that there's something different in terms of the requirement for these two proteins for condensation. Okay, so in the absence of cohesin, chromatin is irreversibly misfolded. So now we can do a subtle twist. We can say, okay, well, what's the reason that this is misfolded? And one idea is that maybe condensin is misregulated. So we did a different experiment. What we did was we depleted both of them, both cohesin and condensin simultaneously, so they could arrive in M phase. They don't have either of these complexes. And now we restore both of them. And what we can see is the condensation comes back. So that tells us that the reason there's misfolding here, in the case when you have cohesin alone, is because condensin is active. If you keep condensin inactive while cohesin is inactive, and you add them both back together, you get proper folding. So in the absence of cohesin, condensin is misregulated, causing irreversible misfolding of the ribosomal locus. So now we've got a new idea that cohesin is a regulator of condensin function to make sure it acts properly. Okay, so how could cohesin be doing this regulation? Well, what we know is that we knew from studies from others that polokinase, a very ubiquitously conserved kinase, phosphorylates both cohesin and condensin. And what I won't have time to show you in this publication is, and I should say 2020 there, is that cohesin is necessary for the proper phosphorylation of condensin by polokinase. And in the RDN locus, binding of polokinase to cohesin enhances polokinase's binding to and phosphorylation of condensin. So that leads to this model. And there's a lot of experiments in that paper that will tell you how we came to that conclusion. We believe that cohesin and condensin both bind independently next to each other in the ribosomal locus. Cohesin then recruits polokinase and then transfers a polokinase to a condensin. And now condensin gets properly phosphorylated in the right places to activate condensin and generate the proper folded structure. So I'm going to tell you, I think, like one sort of killer experiment that shows that this is the case. So if it's true that the role of cohesin is simply to recruit polokinase to make sure the condensin gets phosphorylated the right way at the ribosomal locus, then perhaps we could recruit this polokinase to the ribosomal DNA by something other than cohesin. So what we did was we generated a Cas9 that was targeted to the ribosomal locus, carrying these polokinases called CDC5. And we targeted it to two places. We targeted it to the normal place where it's recruited with cohesin, and about 500 base pairs away at another site. So this is the region where cohesin and condensin overlap in their binding. This is the region where there's just cohesin alone. And then what we've done here is we depleted the cells of cohesin, so this polokinase can't go to the locus by itself, or we guided it to the ribosomal locus with one of these two guides. So you can see in a normal wild-type yeast, about 90% of the ribosomal DNA is condensed in our synchronized cultures. If we don't have any cohesin at all, we destroy condensation, as I've shown you in the images before. If we guide the polokinase to the wrong place over here, condensation doesn't occur. But if we guide it to the right place, now all of a sudden we can restore condensation. Not completely, but actually I think remarkably well, considering it's unregulated and maybe not perfectly situated on the ribosomal locus. So this shows that the purpose of cohesin is to bring polokinase to the right place so that it can, we think, phosphorylate condensin. Okay, so that's great. You can say, well, that's a cute little yeast story. Is it a model for anything else besides the ribosomal locus in budding yeast, which sounds like a pretty bizarre little obscure phenomenon? And what we began to piece together was that what we knew is that these combination of three proteins, cohesin, condensin, and polokinase, were not only present at the ribosomal locus, but by techniques like CHIP, they were also present around the budding yeast centromere. And but budding yeast centromere is only 20 kilobases in length, and it's much too small to reveal in any kind of cytological assay. However, extracting these three things did together was to generate a thin line of condensed ribosomal locus shown here. They were likely to form a thin line of condensed region around the pericentric region of budding yeast. So that was kind of cool. And then me being a very old person made me realize that this actually we had to go back and look at those structures I told you about before. Because there is in mammalian chromosomes, and in fact all chromosomes, a thin line of condensed DNA around the centromere of all organisms, this so-called primary constriction, which was the old-fashioned name for it. Okay, and it was also present at the ribosomal locus in many of these organisms, and there it's called, in classical biologists back in the 1900s, called this a secondary constriction. So that was good. So that made a prediction that in fact these structures in mammalian cells require cohesin to form. Now I told you that wasn't the case. That Tatsu Hirano a long time ago had asked whether cohesin is important for condensation in budding in mammalian cells and hadn't seen any role of cohesin. But he was fooled because what he was doing was he was looking in his reactions at just the fact that you could form these cigar-like cylinders. He didn't ever pay attention to the fact whether or not he was forming the primary constriction. So I asked him to go back and look at his micrographs and say, when you deplete cohesin, do you see a primary constriction? And the answer was, you don't. So now we've got a completely different view when we look at that original micrograph, which is that in budding yeast and mammalian cells, the way you spatially condense chromosomes along their length and change it is to recruit cohesin, use cohesin, and the recruitment of possibly polykinase that's yet to be detected in human cells to generate a differently kind of condensed chromosome. Okay, so that's the first story. The second story is sometimes structure is incomplete. And this is the famous example of the 6 blind men and the elephant. Everybody thinking they understand how the elephant works because they're grabbing a different part. So in addition to chromosomes, these SMC complexes thought to be tethers to hold different parts of DNA together, to either hold the sister chromatids together or to hold a loop within a sister chromatid to generate condensation. Some recent studies, really pioneering studies from the Herring and Decker labs, showed something we had embarrassingly missed, which was that SMC complex were actually loops, complexes that could form loops along many kilobases in vitro. So this is just exhibited here in one of their beautiful in vitro reactions. You have a piece of DNA that's tethered to a slide, where the bright spot is where the SMC complex called condensin, it could be cohesin now, they behave the same. And what you're going to see is over time is that the condensin starts to move down this way, extruding a piece of DNA this way, which is actually a loop that's just collapsed on itself, and you can see it in other micrographs. And so as the cohesin migrates this way, it's actually translocating along these two pieces of DNA and extruding out a loop behind it. Oh, okay, and work from the Rudner laboratory had shown this from bacteria suggests that this happens in vivo as well. So now we have two models for how these complexes work from their structure. If you look at the EM structures of these SMC complexes, they can often look like a big ring. And so one idea is the way they tether things together, pioneered by or strongly postulated by the Naismith lab, is that they embrace the two pieces of DNA molecules between them, thereby holding them together. Now, when we see that they are actually translocases, in fact, this kind of binding doesn't work. Instead, in some recent AFM beautiful pictures from LaRue and Decker, what we now see is it looks like the DNA binds to the two opposite ends of this complex. And then the complex can essentially act as sort of a scrunch or pump by pulling the DNA together, transferring this piece of DNA that's up here to this guy, and then going out and grabbing another one. And you can actually, in that mechanism, possibly generate a loop. So now we see that this original structure, which looked like just a big ring, in fact, was maybe misled us into the simplicity of its function. And it's actually doing something more complex. So now this is where the field is trying to resolve these things. And what I want to tell you is that just like the blind man and the elephant, there's yet another component of the cohesion structure, which is missing from these pictures. And this is the work of a very talented postdoc in the laboratory, Xiheng Zhang, who basically shows that cohesin is oligomerizes, and so that which is a big missing feature of the cohesin. So there was evidence that cohesin may form oligomers. There was genetic evidence from my laboratory in which we found that 2 point mutations in the same subunit, each individually act as null mutants appearing to eliminate cohesin function. The protein is still there, but it appears to be completely non-functional. If you co-express these two protein complexes, proteins in the same cell, you restore function. Since there's only a single SMC subunit in each complex, the only way these two mutant alleles could actually talk to each other and restore function is if the two mutant complexes, one with one SMC subunit and one with the other mutant SMC subunit, came together. Now there is some biochemical evidence also that from single molecules in recent studies, which sets the cohesins in early years in vitro. So this was indirect genetic information. So the question is, do cohesins really oligomerize in vivo? And if so, what's their function and their regulation? So we decided to take advantage of this technology called proximity-dependent biotinylation. And what we did is this assay involves the BRA enzyme, which will take biotin and covalently attach it to an ABI tag if the ABI tag can get into the enzymatic site, active site of BRA. And so we end up with a biotin conjugated ABI tag. If the two SMC complexes get close enough together to allow that to happen. And what you can see here is that how do we detect this biotinylated ABI tag thing? It's very simple. We take the cell extract and run it on a denaturing protein gel. And the SMC subunit, if it has gotten a biotin tag from the adjoining BRA enzyme, if you have in that protein gel strepavidin will bind this biotin tag and actually cause a band shift while the complex is running. And so you can see that. Here's the unbiotilated protein. Here's the biotilated protein that's been band shifted because of the strepavidin. And what first thing you want to show is that this assay is specific. And so what we can see here is that what we have is the differentially tagged proteins. And what we can see is that we only generate this biotylation in trans if there's an ABI tag and there's biotin present. And now when both of those things are present, what we can see, well, the BRA has to be present on the two tags have to be present on the two SMC complexes, and we have to have biotin added to the media, and now we can get this band shift. Okay, so now we have an assay. What this told us right off the bat was, in fact, that there were oligomers, or what we like to call clusters, being formed inside the cell. We can next ask the question, well, let me add one more point here, which I won't show the data for, but we had the ABI tags placed in many places in the SMC3 molecule. And from that, we could tell that the BRA on one SMC3 molecule in one cohesin complex could biotylate the SMC3 ABI tag in multiple places along in the other cohesin complex. And the only way that can happen is if the complexes are highly dynamic in their structure, allowing this BRA to sort of move and find an ABI tag in a different place in the same molecule. And so we definitely show that they're structurally very dynamic, and that may fit with their ability to generate this loop extruding activity. We then look to see if they're cell cycle regulated, and what you can see is that you don't see any of this band shifted ABI tag in G1 cells. In S phase, we see that about 50% of the total SMC protein is band shifted. And that actually comes close to, you remember that the BRA can only biotylate an ABI tag. Another complex just has an ABI tag in it, and only half of the total complex in the cell will have the BRA, and half of the cohesin complexes will have the ABI tag. So the maximum amount of biotylation to expect, if in fact every cohesin was in an oligomer, would be 50%, and that's basically what we see. And then what we see in metaphase is about 50% of that oligomers goes away. So we can then say, okay, we have a lot of oligomers, it's cell cycle regulated. Where are they on the chromosomes? And so the way to do that is we do our biotylation reaction, but instead of purifying, just adding TCA to see how much cohesin's been biotylated, what we do is we do a chip experiment in which we're precipitating the cohesin oligomer on DNA using the strepavidin beads. And then we can detect the amount of this ABI tag associated with any particular DNA sequence by qPCR or ChIP-seq. And so what was known about cohesin binding to chromosomes before this? Well, what we knew was they were highly enriched at the kinetochore in all organisms, and they were at cohesin-associated regions along the length of the chromosome. And this actually was demonstrated by Shika when she was in my laboratory, and this word cohesin-associated region, or CARS, was the definition that she gave this to the field. And so what we can see here is that if we look at these graphs, and we're just going to look at a couple of random places. So we look in the pericentric region, and what we see is that this is the amount of cohesin that's present at that site when we're looking at total cohesin. In red is the amount of oligomers at that site, and gray is control, showing what happens if we don't have any biotylation and the negative control, showing that this red is actually relevant. And we can see that at CARS along the genome, there's a similar thing where we actually can see that there's oligomers at all these sites where we had previously identified where cohesin binds the chromosomes. That's in S phase. In metaphase, what we see is the cohesin remains in a oligomer state near the centromere, but goes dramatically down along the chromosome's arms, in some cases disappearing, in many places going down several fold. So this indicates that there's a spatial control in the cell where oligomers are. So in S phase, what we think is it looks like is that you have oligomers at the centromere and along like the chromosome arms. And then in M phase, what happens is the oligomers get specifically taken apart at many sites along the chromosome arms. So what protects, why don't these oligomers at these sites go away? Is there anything that controls this oligomer state inside the cell? And so we knew that there was a bunch of regulators of the cohesin complex. One complex was a cell transferase. Another was a complex that was known to be important for cohesin loading. And a third protein called PDS5, which was known to be required to maintain the sister chromatic cohesion from the time the cohesion was made in S phase until M phase, when you want to dissolve cohesion to allow cells to go through mitosis. And what we found was that none of these factors affected the ability to make oligomers. But in fact, if we knocked out PDS5, that protection of oligomers that we saw normally in the metaphase chromosomes went away. And so that's shown here. So here's PDS5. And it turns out that it's not required to maintain oligomers, shown by this band in S phase, but it is required to maintain oligomers in metaphase. So now what we've established is biologically there are oligomers inside of cells, it's cell cycle regulated, and we've actually identified a factor that's important for that regulation. So a cute trick you can do is to say, well, okay, if this is truly involved in the maintenance of oligomers and therefore the maintenance of sister chromatic cohesion, do we have other mutations in the cohesin complex itself that are defective for the maintenance of cohesion? And if so, do they affect oligomerization? And it turned out that turned out to be the case. So now we have a very tight correlation. The rare subset of mutations in yeast that affect the maintenance of cohesion also block this ability to oligomerize. So we're excited by this. We think that why would you need to preserve these oligomers at around the centromere in metaphase chromosomes? Is because we know that what happens is the microtubule attach to the kinetochore and try to pull it apart. And it's by virtue of the fact that they can't pull the sister chromatids apart that you generate the mechanisms that help ensure that the two sister chromatids are oriented to go to opposite poles of the cell and therefore segregate properly. So in order to generate tension, as it's called, you need the cohesin to be strong enough, generate strong enough cohesion here that you can keep the sister chromatids together. A second place where you would need this kind of tight cohesion is in meiosis, where the cohesion is part of the process that helps keep the homologs together to establish tension between the homologs to also allow it to get the bipolar movement of the homologs in the first meiotic division to go to opposite poles. And in some beautiful experiments we just found out, again, not knowing in advance, but subsequently from in Villanove's lab, it turns out indeed she has evidence that this part of the genome, actually right here, has oligomers of cohesin. So once again, this observation that we started out in budding yeast now seems to be general, not only general to mitosis, but possibly to meiosis as well. Okay. So I'm going to return now. Not quite sure what that slide's there. Okay. So yeah, that should be there. Okay, so let's turn to the third vignette, which is that structure is irrelevant. So this is very, this is really, I'm being irreverent, and because how could structure be irrelevant? So I want to give you a couple examples. The fundamental premise I'm going to tell you is that sometimes function drives the formation of irrelevant structures, and that we get fooled because we think they're relevant. So in classical cell biology, you know there's a Golgi stacks. And that's something that's been around for a long time. What we now know is that in budding yeast, there are no Golgi stacks, yet they had Golgi. So you say, well, is this some fun, funky thing? Well, you can take a mammalian cell, you can treat it with berfeldin A to destroy the Golgi stacks, And then you can wash out the profeldin A, and you can ask how quickly do they in fact start secreting again. And they start secreting long before you ever see the formation of the Golgi stacks. So whatever the Golgi stacks are doing in mammalian cells, it's not, even though it's so brilliant and beautiful and people have models about transferring vesicles from one to the other, it's not some essential feature of the secretion process. It may regulate it to some extent, but the structure in some sense may be just a consequence of a rate limiting step, which is if you slow down exit from the one particular vesicle and allow entry to go, it will form an elongated structure. And a more sort of trendy topic are these phase separated condensates, which is very trendy these days. I have to tell you, I'm a skeptic. I think they are these things actually functionally relevant? I think is the biggest question in this field. And I think there's very little evidence to suggest they are. They may be the consequence of a protein of low affinity interactions, which are important and generate specific structures. But when you have a lot of that subunit around and none of the substrate, they form these large phase separated liquid droplets. So let's think about now that with that as an intellectual framework, we think about the visualization of loops. So loops are visualized in cells through this beautiful technique developed by Joe Decker's lab. And you have a cell and you cross-link the nucleosomes together in the entire cell. You just add cross-linker. If any nucleosomes are close to each other, you can cross-link those nucleosomes. You then extract the chromatin from the cell, and you digest with a restriction enzyme, chromatin digestion, and then ligate together. And if the two nucleosomes are held together, then you'll generate a chimeric ligation product, which has the two DNA molecules which are associated with those two nucleosomes. You can then do paired end reads, and you can sequence the two ends, and you'll get chromosome coordinates of the contacting nucleosomes. So let's see how that you can then essentially plot that to get some idea about structure. So in this particular case, you have an unstructured piece of DNA, a chromatin. You cross-link in general the neighboring nucleosomes because that's the closest one. And then what happens is you plot the nucleotide sequence, which is associated with this nucleosome and that nucleosome. They're very similar to each other. Let's say this is at position 1000 of chromosome 3. This one will be at 1200. Those two coordinates end up being very close to each other and will lie along the diagonal if you plot them with the chromosome coordinates of two paired end reads here. In contrast, if you have a loop, what happens is you cross-link the two chromatins, but they come from distal parts. So this one now is still at 1000, but this one comes from 2500. And so what happens then is those two coordinates lie off the diagonal. So with that kind of approach, you can look at these 3C maps and get an idea of the types of structures you can generate. So if you can get something which is at the base of a loop, you can get something with the coordinates pointing out there's something called TAD or topologically associated domain, or you can get something which is both a loop and a TAD. And what was found first in mammalian cells was that cohesin was required to make these TADs. And later, even in some cases, in other organisms, it was a condensate-like complex. So this both told us that cohesin not only could tether together sister chromatids, so between two different molecules, but within a DNA molecule to generate a loop, something which we had sort of thought the condensate could solely do. So what were these loops and TADs doing in mammalian cells? And so it became a really elegant idea was that, of course, in mammalian cells, you have a problem that enhancers have to talk to their proper promoter. And if they don't, what happens is that, so in this case, an enhancer here, number one, has to interact with promoter number one to make sure you get proper expression in the heart. And here, enhancer two has to talk to promoter two to get this gene to be expressed in the kidney. And so what they found, basically from a series of experiments, was that in order to form these kind of TADs, you required cohesin, and you required a factor called CTCF, which is just a protein shown here in purple, which maybe helps position the cohesin or actually stops the loop formation. And that they found was the presence of these TADs correlated with gene expression. So if you had a rearrangement that messed up the TAD, it also messed up gene expression, and in case thought to, at least in mammalian cells, causes polydactyly in mice. Loss of cohesin or CTF changes the expression of 1,000 genes in the human cell line. Inactivation of the dosage compensation complex causes loss of dosage compensation in worms. So there's this phenomenon where this is a cohesin-like complex recruited to the X chromosome to downregulate the expression of genes on the X chromosome, again implying that maybe the loops are controlling enhancer promoter interactions. And what made even more sense is, well, when they looked to see whether yeast had these kind of TADs, and it turns out that they didn't. They lacked, first of all, if you do this, here's a picture of one of these, what you would see if you do one of these contact maps, and you plot them out, and you can see these nice spots in mammalian cells indicating they're off diagonal. This would be the base of a loop. This would be the base of another loop. And what you can see in budding yeast is you can maybe see one here and there, but they're very hard to see compared to human cells. So yeast was very bad at making loops, well, the thought was, and not surprisingly, because it lacks CTCF. And biologically, this made sense because in yeast, the enhancers and promoters are very close together. So you wouldn't really need a mechanism to make sure an enhancer talks to the right promoter, for example, by making a loop, because of the fact that they're already going to preferentially bind to the right sequence. But that has changed over time. So that was all the trendy beginning. And what we now know is the rearrangement that we thought was so important, well, it did affect the TAD, but of course it affected a lot of other things. And more careful experiments realized that it was a big inversion of sequences that affected the gene expression, not the inactivation of the TAD. Furthermore, I told you there were 1,000 genes that were affected when you got rid of the cohesin and the CTCF. In fact, the loops in mammalian cells encompass the whole genome. And there were 7,000 genes whose TADs were destroyed or badly affected, whose gene expression was totally unaffected. And finally, we have inactivation of this cohesin-like complex causes loss of dosage compensation. I told you about that. And some really elegant experiments done by Barbara Meyer. What she did was she said, okay, I'm going to go in, and instead of doing this gross stuff, I'm going to knock out all the loops that are being formed by this complex by CRISPR, knocking out at very specific places. Well, the most, and what she found out was she eliminated the TADs and the dosage compensation worked perfectly fine. So now we're a little bit of a conundrum. What are these loops doing? So this is the last story. What I can tell you is that we looked in, investigate the question of whether yeast forms chromatin loops. And it began with a collaboration with Lorenzo and Rebecca again, with Stanley Zai and Xavier Darzak. And Stanley, what he had done was develop a better version of this 3C method, in which instead of using restriction enzymes to digest the cross-linked nucleosomes, they use microcyclic chromatin digestion, which cuts the DNA into dinucleosomes, or cuts the chromatin into dinucleosomes. And then what you can do is purify at least the dinucleosomes, and then what you see in the end, you can do the paired end reads. And so now you're getting ligation products, a chimera molecule, which are very close together, gives you very high resolution of the two DNA coordinates of the contacting nucleosomes. And when he did that, you can see the map here. Here was the mammalian one we saw before, and now here's the yeast thing looking at before. And now what you can see is this is looking a lot more like the humans. We have these off-diagonal spots in the human cells, and we see the beautiful off-diagonal spots along the length of the chromosome here. Furthermore, what you can see here is that this is the effect of a mutant in human cells, the so-called WAPL protein. And what it does is it allows you to expand the loops where there were no spots before, very distal. So these are bigger loops being formed between this sequence and a sequence here, as opposed to this sequence and a sequence there. What you can now see is the abscess protein, what appears to be is this loop extruding activity is more processive, goes longer, and forms much bigger loops. And what we can see is that we knock out the ortholog in budding yeast, and we get exactly the same thing. And I'm not going to show you here, but if you knock out cohesin in either of these organs, you lose all these off-diagonal spots, whether it be yeast or humans. So now we're seeing, wow, yeast looks exactly like humans. We have spots, loops being formed throughout the chromosomes. They're being regulated by the same molecules. And so I think this is really exciting for the field, because I think Yeast is a model to understand the mechanism of looping. As you all might know, it's a lot cheaper to grow yeast in the laboratory. Oops. And so we can start to do it, and we have very powerful genetics. So in my laboratory, for example, we have mutants that upregulate and downregulate the ATPase that's associated with these complexes is likely promoted for its being able to translocate along DNA. And we can ask how this affects loop size and how it affects biological function. Furthermore, we believe that those oligomerization, we have these oligomerization mutants that I told you about before, and those might be very interesting because we think that these kinds of mutants, along with mutants that regulate the ATPase and binding yeast, may be the way that you control whether one of these cohesin complexes is a tether or whether it's a motor that translocates along DNA. So that was very nice. So we definitely have a lot of promise in what we can do with this system, but I want to tell you one example of how we've learned something new. So this is a kind of typical contact map you might see in a region where you get these complex patterns of spots. So this sequence number 1 is forming a loop with 2. It's forming a loop, nucleosome at position 1 is near nucleosome at 2. It's also near nucleosome at 3. And it's also near nucleosome at 4. So how do you get that happening? Well, if you imagine this is kind of the cross-linked view, this nucleosome in 3-dimensional space is near all 3 of these nucleosomes and could be cross-linked to any one of them. And that could give you this pattern. And then what this envisions is that the folded chromosome is a quaternary structure with some defined features like as shown here. So what we noticed in our yeast system was something, because we had such sequence depth and we get very good resolution, is that if you look at where these spots are, these off-diagonal spots, you can correlate them with the position of where cohesin binds the chromosome in experiments done by Chipsy, exactly like Chika did many years ago. What you can see here is that here's a cohesin at this position, and it seems to pair with a cohesin at the neighboring position about 15 kilobases away. And you can see that over and over again, where the next one down pairs, the third one pairs with the fourth one, and we can keep going down. The fourth one pairs with the fifth one, et cetera, et cetera. So this implies that the 2 regions are brought together by cohesin. And we also notice that the intensity of the spot seemed to fit with how much cohesin was bound at the spot as determined by ChIP-seq. So we know there are 2 populations of chromatin-bound cohesin. There's a dynamic population, which is presumably doing the loop extrusion. And there's a stable one, which we think may be actually generating the cohesion, or at least the precursors of forming the cohesion. And we know the stably bound cohesin is at these sites. Prior to loop formation, we established that through kinetic assays. And so that led to this following model. We have stably bound cohesin at these sites. A loop extruding cohesin gets on and starts making a loop. And the loop keeps going until these dynamic guys run into the stably bound cohesin and then they stop. So this is a new mechanism for how organisms could generate specifically defined loops just through the binding of cohesin itself and its looping activity without the factor like CTCF. And I should say that in many organisms, CTCF doesn't exist. And even in mammalian cells and others, there are loops that are CTCF independent. This may be the underlying mechanism for how you generate a loop. So what we can see now is that this kind of mechanism allows you to get a very different way of generating these complex maps. Because what we said is, here we have the intensity of where the cohesin is binding along the length of the chromosome differs. And so we can see at this particular site, it's present on every DNA molecule in every cell of the population. That gives you a very high ChIP signal. The next site over, it's stochastic, and in fact, during the DNA replication, it was only put at two of these sites. 2 of the 4 DNA molecules at that site had it. So now we're going to get a lower ChIP signal, and you can work with it. Now you add the dynamic cohesin that's generating the loops to these different molecules inside of different cells, and what you're going to see is you're going to get a different pattern of loops in each cell. So each cell actually has a different pattern of loops. It's not that all cells have the same quaternary structure, but they have different patterns of loops depending upon the stochastic loading of the stable cohesin. And when you add all those patterns up and you look at them as a population, you get the exact same pattern you saw before. So in this way, we think that in some ways, the loops are quite diverse. And now it becomes hard to imagine a model of somehow about regulating gene expression, because in a tissue like your lung, you want all the cells to be expressing the right kind. You can't have a loop be, sometimes a loop be there and sometimes a loop not be there. So those combination of things, it's in yeast where we don't need enhancer promoter interactions. It's all over the place on all chromosomes where many genes aren't being regulated. And it is sort of possibly different between individual cells. So that brings up the question is, what is the function of loop extrusion? It doesn't really fit with it being a global control of gene expression. It might be important for a subset of genes, but doesn't seem to explain it. So we can look to the biology of the extreme. We can look at immunology. And there's some beautiful papers done there where it shows that what the cohesin complex is doing in that case is controlling antibody class switching. It's controlling the recombination reaction by juxtaposing the right sequences, by shuffling the sequences to generate the right, bring one right run in proximity to the other. So in this case, the structure is actually sort of a downstream product. What the real function of these proteins are is to shuffle DNA sequences so that they end up getting next to each other, not the final structure of generating a loop. And so I want to leave you with this idea that what could that mean? Well, we have a thought that maybe this would be what could be the global function conserved between yeast and human cells that these loops could do. It might be repair of damaged chromosomes. So imagine you have a break in a chromosome and it happens to be in a repeat. What you want to have happen, if you want to have the two sister chromatids remain the same, is you want this break to be repaired off of repeat number one. And that if it's true, you'll put back the sequence and it'll be fine. If the break in the first repeat actually uses 2 as a template, 2' as a template on the other sister chromatid, you have the possibility of having a reciprocal recombination event, which can generate duplications and deletions, and that's not good. So now what we can do is imagine, though, we get a looping enzyme comes on here and it starts looping. What it's going to do is going to suck these 1 sequences in, and it's going to bring the broken 1' close to the first sequence. And that will help direct the proper reaction. So it's maybe a way, particularly in very complex genomes like humans, where there's lots of repetitive DNA, to control the proper recombination to keep sister chromatids intact. So the take home from this whole talk is that there's temporal and spatial regulation of condensation along the lengths of chromosomes. We think we found a mechanism that's conserved from yeast to humans. Cohesin oligomers or clusters are regulated temporally and spatially to control tethering and possibly loop extrusion. And finally, chromatin looping is conserved function when yeast to humans, suggesting a more functional fundamental role than the sort of the more highly touted current idea of controlling gene expression. And we suggested one possibility in DNA repair. And so I'll end my talk there with thanking all the people in my lab. I've mentioned the people who did the work along the way, Rebecca and Lorenzo, and Vincent Guacci shown here. And I stop and answer questions. Thank you.
Thank you, Doug, for this excellent presentation. So I think we do have some time for questions. I will be asking some of the questions on behalf of the audience members who have been posting the questions in the chat box. So one of the questions that we have for you is if yeast cells lack CTCF, so what drives the anchoring of the loaded cohesin that extrudes the loop? And another part of that question is, are TADs always involved in gene regulation, or do they have non-expressional roles and might just be involved in condensing the genome? Your views on that?
Okay, so oops, go back here. Yeah, so our vision is that the function of CTCF has been replaced by a second function of cohesin, which is that there are really 2 populations of cohesin. So the idea in mammalian cells is that cohesin binds to someplace along the genome and starts generating a loop. And it comes along until it runs into CTCF. And when it runs into CTF, CTF has a little stop sequence that says, cohesin, you have to stop here. You can't go any further. And our view is that function of CTCF is taken over in budding yeast by just stably bound cohesin. So the motoring along cohesin comes along and it runs into a cohesin which has been modified to not be a motor anymore, but modified to be stably bound at a place so that it can become cohesive, a tether instead. And when it runs into that, that's how you stop the motor from running down the length of the chromosome. I hope that's clear. So that's the idea. And we actually think there's a beautiful from Dan Gerlach's lab in mammalian cells, which also suggests that loops stop at sites of sister chromatin cohesion where there are sites of stably bound cohesin. So we actually think that that's we may have found the same kind of activity there. And then, yeah, as I sort of alluded to at the very end, we think that the function of the looping could be in DNA repair. It could be some sort of function in compaction. That's also another possibility. I don't think we can eliminate that at the moment. It's certainly a possibility. And we have found, well, it's still, in mammalian cells, even in the organisms where you, there are examples when you manipulate cohesin outside of budding yeast and where you change the level of condensation, even on the parts of the chromosome that aren't the pericentric that I told you before, implying that while it's not absolutely essential, as Tatsuya Arano has implied, it actually does regulate the size of the loops. And that could be either because itself is controlling the loop size or it's indirectly affecting condensing.
Okay, I think that's a good answer. So the next question we have for you is CDC5 not required for condensation in chromosome arms, or is there a cohesion independent mechanism for that? And also whether you know whether the phosphosites on condensin are known. So the first one is CDC5 required for condensation.
Yeah, we have not, we've gotten, so I'll say to you that in all the sites where we see cohesin bound along the chromosome arms, the kind of sites that Chick identified, polokinase is at those sites as well. So it doesn't only localize to where the cohesin is in the ribosomal locus and the pericentric region. Cohesin, CDC5 is present at those other spots as well. The difference is that condensin is not present at those places. So the only place where you see both cohesin and condensin is around the centromeres in the ribosomal locus. But that doesn't mean that polokinase, it could be there, and we need better methods. And we have not directly assessed. We can look at the condensation of the non-ribosomal DNA by more complicated methods than our simple cytological assay I described here. And we haven't done that. It's something we should do for sure. And then the second question was the second question about
the location of the phosphosites on
condensin?
condensin, is that known?
Damien Demore's lab has identified a lot of the phosphorylate. There's a lot of them. Polokinase phosphorylates many sites on many of the subunits of cohesin and condensin. So that's why we haven't pursued finding the specific phosphorylation sites that might be the ones regulated here. But what we do know is that you do see a difference in the, you can look at the sort of a crude assay about the phosphorylation sites by the band shift when you phosphorylate something and it migrates more slowly in a gel if it's been phosphorylated. And we see that phosphorylation occurs in the absence of cohesin, but the phospho shift is very different. In fact, it looks like it's hyperphosphorylated. So it's clear that you don't need cohesin to get condensin phosphorylated. You need cohesin to get condensin properly phosphorylated. But we don't know at the specific level what that is. We just know there's a gross change in the pattern of phosphorylation when cohesin is present or compared to when it's absent.
All right, so I guess the next question is whether one of the members of the audience wants to know whether different chromosomes also interact with each other, or do they have any kind of other links or connectivities, different chromosomes?
Yeah, there is. I mean, if you look at the these chromatin maps, these contact maps, they visualize any contact that's happening inside the cell, and they can, done properly. And they do see interactions between, so in almost all cells, whether it's humans or yeast, the centromeres or kinetochores are clustered together. And that's an old-fashioned thing from the Rabel conformation. There may be more specific proteins that help mediate those interactions. So that's one level of interactions you have. There are also, I think, bigger ideas of big domains of certain proteins and different chromosomes interact. But we don't know anything about that. We don't know how much cohesin and condensate could contribute to those. It's an interesting, just point out, it's one of, I think, one of the big unanswered questions when you start out and you think, oh, this is a very simple problem. You have a complex, and in some ways I've drawn it, a protein complex. It grabs onto two pieces of DNA together and tethers them. But if you put that in context of the nucleus, you can understand that those proteins, because they can act at multiple places along the length of the chromosome, could easily cross-link two different chromosomes, as well as cross-linking two sister chromatids to make a sister chromatin cohesion, or two parts of the same sister chromatid generate a loop. So you can imagine these things would just end up cross-linking the whole nucleus into a big mass of yuck. And somehow the cell, these complexes know what to do. They know who they're supposed to cross-link, and not just randomly generate cross-link between any two neighboring pieces of chromatin that you would have in an interphase nucleus.
All right. Another question that has come is there any kind of quantification of the amount of chromatin-bound cohesin during interphase versus mitosis?
Yes.
Yes.
There's a, well, what we see and other things, there is, there's not much of a difference between the two. And I probably, in the cartoon I drew was incorrectly, which is that I should have made the point that the difference between S phase, where you have oligomers and where you don't, is not that there's less cohesin, but they're just not together as oligomers. So it's, if you imagine, let's say in a normal mitosis, or at one particular site on a chromosome, you have an oligomer that looks like this, where the two are stuck together. And we see that by our experiment. What I'm suggesting is that what happens at other sites is there's still two molecules there, but without PDS5, they're no longer not stuck to each other anymore. And so there's the same amount of cohesin there, but you don't have an oligomer.
Okay, there are a lot of additional questions, so let me sort of go through them fast. So one of them is, does the anchored cohesin bind to specific sequences like CTCF? If yes, this shows sequence specificity of cohesin that is not known. So does cohesin directly bind there, or could they be potentially anchored by some other protein similar to CTCF?
That's
similar to the earlier question.
So the answer is there's yeast has been very heavily studied and annotated, and there are no other DNA binding proteins that have a chip pattern that's identical to where cohesin is. So we don't have any evidence that there's equivalent to anything like CTCF in budding yeast. We can't exclude that. That's certainly a model that we're missing the thing that's helping it to determine what it is. The sequence specificity for cohesin is controversial. What's known is that there's a strong correlation that where cohesin is in budding yeast and is between two convergent. It's here, if the transcription is this direction between two genes, you tend to find cohesin at this position. And that for a long time was interpreted by the fact that cohesin bound anywhere, but as transcription went along, it would push these rings of cohesin down the chromosome to the end of a gene, but it couldn't keep going if there was transcription coming from this direction. So it was kind of like the idea of, you're basically going to push cohesin down like that. It's all going to fall into place between the two of convergent transcription. I personally have never liked that model. So that says, well, what could be the unique feature that's causing them to go to that site? In vitro experiments that we did, cohesin does seem to prefer highly AT-rich DNA, which would tend to put it at the ends of genes or maybe the beginning of genes. That's one feature. The second thing, which I've always entertained, which may not be true, may or may not be the case, is that it is that it may be some structure that's formed at the DNA as a result of the convergent transcription. So you may actually generate a loop of DNA there because of transcription, or maybe there's an RNA associated with it. And those are the kinds of things. But it's something I think we need to go back and visit now to understand exactly how cohesin knows to go to those sites. And we don't know.
Okay, so there are a large number of questions. I'll actually go through them in serial order rather than.
I'll be quicker in my answers here.
Yeah, there are a lot of them, maybe more than 10 questions. So
Okay.
I'll quickly ask the next question. Do you think there's a role of WAPL in oligomerization in addition to PDS5?
Yeah, that's a good question. We eliminated it, and we don't have any. But at this point, it doesn't seem to affect oligomerization.
Okay. How do you envision loop structure maybe in yeast RDN1, which is highly repetitive?
Yeah, that's a good question. I didn't go into that. You can look at the paper. It's been published now in eLife. We see a specialized contact map in the RDN, but it's very difficult to interpret that map because any paired end read you get between two sequences could be those sequences within a repeat, or those one end is in one repeat and one end is in the neighboring repeat because it's a single series of repeats. It's always the problem you have with repetitive sequence. How do you know where the sequence is coming from? Is it coming from the same repeat number 1 and 2, 1 and 3, etc.?
Yeah.
So we don't know how to interpret that at the moment. It's a good, we might have to put in some polymorphisms to be able to test that issue.
How does cohesin select binding sites? I guess it's related to the earlier question to form
Yeah,
tags,
again,
etc.
it's
Not so clear.
Not so clear at this point. Nope. Shika laid that question out many years ago, and nobody's answered it yet. So it shows you anyway.
Yeah. During meiosis, oligomerization of cohesin requires temporarily at predefined sites because if oligomerization happens at predefined sites, then crossover will be happening at the same locus. Please clarify.
Yes. Okay. So again, that's something we don't quite understand, but there are two subtypes of this experiment was done in worms. There are two subtypes which have different. One subtype of one cohesin complex has a REC8 subtype, and the other complex has something called CO3 and CO4. They exist all over the chromosomes, and then in early stages of meiosis. But then at the stage where you have pachytene, or you've done the recombination, what you find is that this CO3, CO4, which is the one that forms the oligomers, is only present distal to where there is a crossover. And so the answer to your question is sort of wherever the crossover happens, somehow dictates which oligomers are going to be stabilized nearby. And nobody really understands how the CO3, CO4 totally rearranges in the genome and ends up being just distal to where the recombination site is. But I think it's the causality is somehow where the recombination site is then dictates, maybe puts out some local factor that stabilizes the oligomers there, and other sites where there's not a crossover, there's not a signal set, and the oligomers get taken apart. That would be my model.
All right. So next is looping is a form of structure which seems to be important for function. If yes, why do you think that structure is irrelevant related to your title?
Well, I just think that it's not. It's just it could be. It's a little bit tricky because is it? I guess goes the idea is the looping. You could say, well, you're generating a structure where you brought the two DNA sequences next together, for example, for DNA repair, and that's sort of that is sort of a structure like context. The other way to think about it, though, is that it's certainly not that you're forming a loop to compact the DNA. I mean, you could have anything. You could have it be a loop. You could have it be a TAD. You could have it be anything of that DNA that's being extruded. And that structure is what I meant is not relevant. So whether it's a loop or a TAD or whatever, it's the only important thing is you got these two sequences at the base next to each other. And in the sense that, you could cut the DNA and you would generate no structure and it would still move along after it's been bound and bring the two sequences together. I was being provocative in terms of, and good question.
Okay, so next is what determines a cohesive SMC and what ensures that the other one acts as a stop for loop extrusion? Could it be partners or PTMs?
Yeah, it's a possibility. I mean, what we discovered is, and I know this, is that we published a paper just doing some in vitro biochemistry, is that there's regulation that needs to make a cohesive, and that involves acetylation. And only a subset of the total cohesion in the cell is acetylated. So we think that's the mark. Then we did some biochemistry to show what acetylation likely does is downregulate the ATPase. So the simple idea, the way I think about this is, how can you have a complex be both a loop extruder and a tether? And if you think about loop extruding, what you have to do is you have to bind two pieces of DNA, and then you can do the scrunching reaction, like you say. So it's bind two pieces of DNA, you bring one close together, so if you stop the loop extruding cycle, which is you bound the two pieces of DNA, but you don't allow the subsequent steps to happen, which would loop extrude, then it becomes a tether. It's just the way actually kinesin and myosin can become so rigor binding. The same thing, you stop the reaction in the middle of a looping reaction, it's actually tethered to two pieces of DNA because it wants to go on and make a loop, but it can't do it. And so that's the way we think you determine whether something is a looper or a cohesin, is simply by having this acetylation reaction, which blocks the reaction so that it can't get past the initial binding of the two DNA molecules. And so, so that's, I think, is the person saying exactly, it's post-translational modification. We think in this case, it's a acetylation event, which is determining that. It also could be this oligomerization. Once they're oligomerized, they can't loop anymore either.
All right. The next question is, do you think centromere clustering in yeast is influenced by oligomerization of cohesins? Has anybody tested that?
That's a good question. We haven't had, since we have a mutant which blocks the oligomerization, we could in fact see if the centromeres are still clustered or not, or whether that's a cohesin independent thing. We have not tested that. Good question.
Okay.
Wrong question.
Yeah. Are cohesin molecules recycled? And could there be any role of the UPS pathway in regulation of looping?
Our cohesin molecules, they're definitely both there's a dynamic pool, which is turning over all the time, we think. So we think even in mammalian cells, the loops are being generated and then they're falling apart and they're being regenerated. And that's one form. We also know the stuff that's acetylated is deacetylated and returns into the pool. And I think all that's very important because cohesin is actually, I didn't get into this detail, I sort of focused on one of its functions, but it is definitely involved in DNA repair. It's involved in probably DNA damage in S phase. It's involved in cohesion, as we talked about, and it may be also involved in condensation in certain places. So the cell has to parse all those cohesin molecules to do different functions and make sure there's a pool around to do all the different types of activity. And so I think its regulation actually is going to be very complex to carry out multiple functions inside the cell at multiple different places. And we're just at the tip of the iceberg, and modifications are certainly one way to do it, both of the chromatin and the cohesin itself.
All right, so next few questions are related to loop extrusion. One is there any relevance of oligomerized cohesin with its stable binding at CARs or intransient binding for loop extrusion? In other words, what's the function of oligomerized cohesin for cohesion or loop extrusion?
Yeah, so I mean, again, the mutants that we have that block oligomerization, the PDS5 mutant, can't maintain cohesion. So we think the simple interpretation there is they are essential for cohesion. That we know. We are actually just in the process of looking at, well, I can tell you the mutant PDS5 that blocks prevents oligomers, what we see there is huge loops, very, very big loops that are formed. And that's a little bit of a complicated story, but it certainly implies that, and in mammalian cells also, PDS5 is not required for forming loops. And I don't remember if they know they're bigger also there. So so the simplest interpretation there is you don't need oligomers to form loops, and it's possible that you need oligomers to stop loops. So when you don't have oligomers, the looping activity can go further along the chromosome before it runs into an oligomer, and therefore you get bigger loops.
Another question related to loop formation. Could the number of cohesins recruited control the size of the loop being formed?
Yeah, that's a good question. And I mean, those are all kinds of things. I think now in yeast, we have all the tools to be able to address those kinds of questions. And we're currently in the process of doing those things. So I can stay tuned. And one of the things I found from those studies, which if you can look at the actual paper we published, I think there's like 80 figures, which I didn't torture you through. But it's very complicated, as you know, to answer to. Those are not simple experiments just because you get big data sets, but we're doing them. So we'll know, hopefully in a year, I can give you the answer to that.
Okay, there's a technical question. It's related to BER-A, the assay that you spoke about. BER-A is able to biotinylate proteins which are in close proximity to the tagged proteins. Do you think there must be some other cohesion molecules which do not oligomerize that also get biotinylated? I think there are a lot of questions coming, so at some point we'll have to stop. I think Professor Koshland might be exhausted also.
Yeah, I'm a late night present, but no, I think so. Remember you don't basically you biotinylate. There's an AVI tag, a small subunit amino acid sequence you add to your reporter protein, and that's what the BRA has to recognize to put biotin on. So it's not randomly biotinylating lysines on nearby subunits. It's only biotinylating an AVI tag wherever it's in the cell, and yeast chromosomes, yeast proteins don't have AVI tags. So it's in this sense it's very specific to the particular protein, and the idea is if the BRA can get to the AVI tag easily, that is if they're in close proximity and unconstrained, you get biotinylation. If they're unable to interact, you won't.
The next question is, in the first part of your talk, you showed how expressing polokinase with guides specific to the NTS1 region of rDNA could only restore around 50% of condensation. So could it mean that there could be competition between the complexes for binding at the overlapping regions, which could be responsible for the condensation defect in absence of cohesin?
Get that? I mean, I think there's many reasons. I guess to me, it's shocking that we got 50%. First of all, we think that there is more than one cohesin likely bound to each one of those RDN sites. And with CRISPR-Cas9, we're just recruiting one. The second thing is that, we are changing possibly the stoichiometry. I mean, the whole reaction may be cell cycle regulated, and we're sort of, with the CRISPR-Cas9, we're just bringing it there independent of any kind of regulation, which would also make it less efficient. But there could be certainly competition. There's lots of possibilities why the experiment shouldn't have worked, and therefore the fact that we got it to work to 50% is, to me, a pretty powerful conclusion.
All right, the next question, if you're up for it.
Sure.
Cohesin has been implicated in arm condensation as well. And since these sites are not shared by cohesin and condensin, how does cohesin control condensation at these sites?
At which sites, sorry?
Arm sites.
Oh yeah, arm sites. Yeah, that's a good question. I mean, again, you know, one interpretation from that experiment is that what happens is you know, we need to do the experiment, but you know, the condensate that's at the arginine and the pericentric region is a little bit unusual because it's obviously very it's localized to those spots. It doesn't mean the condensate isn't binding along the arms as well, and because we know that's condensate dependent. So it could be that, you know, the condensate is a loop extruder making a cis loop that condenses the chromosome, runs into the cohesin where there's polokinase, it phosphorylates it, but the condensate kind of moves away for a while before it actually shuts off. I mean, that's the view we have, is what the phosphorylation is doing, cohesin is doing is it, so normally these loop extruding activities, the cohesin condensate, are unstable. They make a loop and then the loop falls apart and they remake the loop. And obviously if you want to condense the chromosome, that's maybe not what you want to do. You want to make the loop and make it stable. So one idea is that what the condensate cohesin is doing when it's polokinase is that when the loop forms and it runs into the cohesin there, it phosphorylates it and shuts the motor off so you have a stable loop. And it's possible at the arm sites, it runs into it and then keeps looping for a little while before it stops working or something. I'm just waving my hands here because I don't really know the answer, but it's a good question, and the model isn't perfect in that sense because we are ignoring that, as you said, CDC cohesin is certainly present on the arm sites and cohesin is required for arm condensation, and we haven't yet figured out why.
Okay, actually there are a few more questions, but we are running out of time. So maybe we can have them clarified later. I apologize to the people who asked those questions.
You can send me an email and I'm happy to answer questions, whatever. Anyway, It's a very interactive audience. I'm glad to hear that there are people out there besides my lab that are interested in these really fun complexes, which I think is, there's still a lot to be learned.
So let me just try to summarize the talk, just wrap up.
Sure.
So thanks so much, Doug, for this informative talk and presenting all these latest exciting findings on chromosome structure from your lab. And we hope we'll be able to welcome you in person someday in ISC.
Yeah.
I thank all the participants who have joined. I thank the team of colleagues, especially Dipankar, Sandeep, Raj, and our chair, Rangarajan, and also Professor Rao for supporting in organizing this event. I also thank the student volunteers. Many students have helped, but to name a few, Shobik, Shitaza, Swagatanath, Mani, and Naren, who have been helping us in this question relay. And I also thank Digits from IC for helping us out in initiating the links and troubleshooting. We hope to have more such events. Many events are coming up for the centenary celebrations, and we hope to continue our interactions in the future. Finally, I thank you all, thank all the participants, alumni, eminent scientists who have taken the valuable time to join with us this morning. Thank you, Doug.
All right, bye-bye.
Bye.